Okay, well, thank you very much for the invitation. I'm sorry, I just had to set it up now because the email didn't go through very well when I said the presentation. So, what I'm going to talk about mostly is not our research. I'm going to talk about some challenges that we're facing in some of the more infrastructural aspects that we're thinking about, both in terms of the Genome Center activities that the Genome Center has been launched last year, and it's primarily serving genomic needs, but also an effort that is being put together to integrate both the experimental as well as the analytical part of multi-omics data, and how that can have an impact on the clinical side as well as on the research side. Now, the problems that we're facing are very complex, and of course, I'm sure you heard from the other speakers, including Mark, who's the last speaker, about how complex the biological as well as the clinical problems are. And we're dealing with a very complex biological system. This is the human body that has dimensions both in terms of space, so different organs, different tissues, different time points and exposures at any given particular time, as well as the time, which is the aspect of time when it comes to exposures, but also time with respect to aging. So tissues generally change as individuals age. They switch functions. They change slightly their differentiation state, and in some cases we have apoptosis and many other biological processes. And not surprisingly, we have a strong environmental effect that has also accumulating effects in time, some of which has a memory in human cells, some of which just passes and goes, leaving sometimes no footprint. And to make things worse, the individual cell behavior tends to be actually very complex. So we cannot just take a piece of liver and do some assay and consider that every individual cell in that tissue is having the same average behavior as we can infer from multiple cells. And that, of course, makes a very complex picture, and one would like to understand how this information is going to be retrieved in a way that, first of all, is realistic, because doing, for example, omics on every single cell in the human body at every given time point means that you actually have to sacrifice the individual at one point, which is unrealistic for humans, as well as actually capturing it multiple times in different parts of their aging process, which of course is unrealistic and impossible. So somehow we need markers, and somehow we need a way to capture that information at a given time and also in multiple time frames. So much of what work that from my lab that actually triggered many of the questions that I'm going to talk about has to do with one way to address a particular molecular phenotype, that's gene expression, transcriptome, that's another way to express it, and to actually link that with genetic information. So for many, many years, we've been doing what is called an expression QTL analysis, or EQTL, that stands for expression quantitative trait loci. So that means that what we're looking for is genetic variants that are actually having an effect on the level of expression of the gene. And the way we do this analysis in a very sort of quick and superficial way to explain the technical aspect is we capture the variability in the vicinity of the gene, where we expect many cis-regulatory elements to be found, and we correlate with the level of expression of the gene, which we normally measure with RNA-seq data, or in the past we used to measure with microarrays. You can do the same kind of analysis with calling trans, meaning that you start capturing these trans-regulatory networks that are very informative, but also opening up the space of a test to a much larger number of nucleotide position SNPs, which leads to, of course, a multiple testing burden, and of course, much lower probability of discovering the same effect size if you were to do this in a more targeted space. So a standard EQTL looks like this. In fact, stratified by genotype. You see the level of expression of individuals that carry a particular genotype changing as you change the genotype. In this case, you can infer this additive effect that you're changing from an A to a 1C and then 2 Cs, and the expression goes up. You will infer that the C allele, the C carrying allele, drives higher expression than the A carrying allele. And this is more sort of a simplistic way that we'll be doing that and trying to integrate all this data. Now the problem is that this transcriptome is just one of the omics, and there's a lot of different omics, and you can call it, the different omics have sort of invaded any kind of data definition that we have. What I'm going to talk about is molecular type of phenotype. So we have two general types of omics, at least from the traditional sense. So this is the quantity based, so this is transcriptome, proteome, metabolome. So these are specific measurements that we do that represent specific parts of the molecular biology of a particular cell population. This is basically the transcriptome, this is the RNA, this is the proteome, this is proteins. You can do this with targeted as well as with more sort of shotgun type of approaches. So we just blindly sample the proteome, and metabolome, where it's a sort of, again, shotgun or targeted for metabolites in whatever sample you're looking at. There's also more discrete types of omics. This is the genome. The genome is basically genotype, so you can have in any position, in a simplistic scenario where we have a single nucleotide polymorphism, you have the 3 different genotypes, and that's discrete information that gives you a different kind of identity. And I'll explain to you in a minute how this makes a difference. And of course, this information can be captured by RNA, by sequencing or SNP genotyping. And then what you can do is you can start linking those two. So you can generate, as we call eQTLs, so the thing that I described before. You can look at protein QTLs, metabolite QTLs, methylation QTLs, and a whole bunch of other things that you can generate molecular data for. So what we can do is now link the molecular phenotype with the genetic information that we have for the same individual. Now, these 3 levels of complexity are actually the 3 levels of complexity that I see from our perspective that poses challenges for this aspect of data protection and identifiability. And I will go into this in more detail now. So you can think of that signal of being able to identify an individual. And again, identifiability comes in different flavors. So I hope I will be able to sort of express that in a more concrete way as we go through that. Is this signal within data? So let's say that I give you a transcriptome, and I'll be able to see, compare, for example, to a normal reference that I have, and that's easy to get because we have a lot of that data, that this individual has, for example, an activation of a particular pathway. That by itself is actually information that could create all kinds of problems with respect to consequences, even for, let's say, insurance companies. Because if you see, for example, an inflammation pathway, you don't have to necessarily categorize that individual into a particular disease state. Already you're calling that individual different from what is sort of the expected normal. So already within data information is posing a problem, and that's just quantities, right? I'm not talking about sequence information, I'm just talking about quantities of metabolites, or quantities of proteome, or even the transcriptome without using the sequence information. The other thing we can do is actually go even deeper. So if I have, for example, a diabetes cohort, or a lupus cohort, and I have transcriptome in blood for those, and also from healthy individuals, I could actually put an individual into a particular class of these reference sets that I have. So I can take an individual, do a transcriptome analysis, and I can put them into the lupus category. So I can actually create diagnosis type of assignments just from looking at omics data. And this actually has been done in many cases. We recently had a study in lupus where we can place with very high certainty, almost 100%, an individual to a lupus group as opposed to a healthy group. And you can start also classify whether they have activated or inactive state, whether they have nephritis or things like that. So this is actually quite strong information. And again, this doesn't involve sequence. This still involves quantities. And the final thing is, once you can infer the genotype from that particular assay. So, of course, I'm not even going into this obvious thing where I have the sequence, because once I have the sequence, I can easily match to another sequence database that I have. But if I have data that allows me to infer some genotypes, even with low confidence, I can actually compare and put that individual into the context of a genotype database, genotype-only database that we have separate. And I'll show you a few examples of how we do this. So we can identify the patient as a case for a particular disease. We can identify the patient as part of a group of patients or a member of a study. I'll show you there was a study a few years ago that actually created a very big issue with the NIH and the availability of data that was a DB gap, because people were not aware of that kind of problem at the individual level. And also we can identify the patient once we go to the patient and we have a specific link with the HRs. Now we can actually treat all this data. And I'm saying the HRs here because clearly we don't have a lot of transcriptome, protein, metabolome data that links this individual to HRs. But we have, for some patients, linked with the genotype data, and we will have more of that in the future. The first assay that enters the EHR environment and links with whatever other clinical data is the genotype data. So if an omics assay proteome analysis allows us to infer genotypes that will link with that HR that actually poses a risk. And in some cases, we can discuss it, actually proteome data can be completely freely available. So actually you can find in databases quantities of proteome data from patients freely available without any controlled access, just because people think that they don't have identifiability information. So here's the paper, this came out in Apologetics in 2008 from David Craig's group, that specifically said that I can actually look at an individual's genotype and look then at the summary statistics of a study that has been done, a GWAS of a particular study, whatever, diabetes, lupus, whatever disease you want, and I can actually say whether that individual was part of that study and whether that individual was part of the cases or the controls. Now that, of course, is not intuitive, because how can you get summary statistics on the one end and just a genotype profile on the other end and make that match? And the reason that you can match this is because you have millions of SNPs with a little bit of information in each, so you cannot make from 5 or 10 SNPs, but you given the millions of pieces of information with a lot of uncertainty, you can actually make the call. And so that means that if I have a genotype and just the summary statistics that are freely available everywhere, so there's a lot of studies for the right reason actually that release the summary statistics, I can actually say whether an individual is part of that study, which is pretty, again, not intuitive. It took me a while to understand how this can happen, but it actually does happen, and with a very high confidence too. The other thing that we can do is we can actually use EQTL data to derive genotype information of an individual. Now, if you look at a certain data set, there's a lot of genetic influence of the genome to gene expression. So if we do, for example, an EQTL analysis with 1,000 individuals, we can discover maybe 10 or 20,000 of EQTLs. That means individual variants that have an impact on expression. Now that means that there's a certain amount of information in the expression level that comes from the genome. And again, I cannot predict the genome just from the genotype, so that means I cannot go back with an expression value and predict the genome itself. But what I can do is I can collect all the transcriptome data, all the expression of the 10, 15,000 genes that I have measured in that particular tissue, and collectively I can derive enough genotype information that then I can match this individual with any genotype database. So I can tell you whether that individual was actually part of a study again, not from summary statistics, but from just having a genotype database. So I can tell you that this transcriptome belongs to this exome data in the clinical records. So that means now whatever information I have here is actually linked with whatever information I have there, which could be anything, AHR or other things. And you can do the same things with metabolites, and this is something we did for a project called DIRECT. It's a type 2 diabetes study looking at even blood transcriptome and blood proteome and blood metabolites. And this is basically the confidence with just 39 mismatches that we have from this, the confidence in which we can assign individuals to their original genotype. So basically that means that I can actually get the metabolome to be generated randomly, and I will make 39 mistakes out of a few thousand, assigning the individual to the genotype that I've measured completely independently. That means I don't even have to keep track of names of samples for the most part, right? And for transcriptome, there's no question about that. As I have the sequence, we actually don't worry so much about writing the names because we can easily match. We have algorithms now that we can match the sequence of the transcriptome, not the quantity, but the sequence with the individual. So there's enormous information just from looking at quantities and also in cases of sequence information. Proteomics, for example, you also have some protein sequence information there. So this all poses a lot of risks and a lot of exposures that we then have to discuss whether it actually starts to make sense to discuss about hiding and protecting data or some kind of regulatory framework. So we can think that, actually I missed a slide, I'm sorry. So the identifiability comes at multiple levels, and what that slide had, because I didn't understand that I was actually meeting a speaker given that there's a speaker that could not make it. So one of the things that we've been discussing within the context of the Genome Center and also the general efforts of multi-omics is to see how we can create a data framework that is compatible both with research and clinical needs. Now, there's all these conflicts that I'm sure have come up in different settings, and this discussion will come in the future talks and discussion, is that On the one hand, you need the data to be protected as much as possible because that's private data, and that poses several risks, partly because we don't have the regulatory framework to protect the patient and the individual. But the problem itself, if you flip it around, is that there's no way you can do any sort of personalized clinical interpretation if you don't have all the detailed information at the same time. So, and the same happens with research. If you want to actually advance research, you need to have the raw information, not the summary statistics and so on. So we ended up in a sort of a vicious circle where we're actually trying to protect the same information that is necessary for the purpose we generated that data. And then it becomes a complete nightmare in terms of building a regulation on that. So the interoperability comes at multiple levels. And we have to actually understand that, and we have to think of the integration framework that we need to apply for this. The measures that we get for that depend on what exactly are we trying to protect. Are we trying to protect the patient from not having that information available so that their friends cannot find out that they have a particular disease? Or we're trying to protect an individual from a legal perspective that they don't get a higher premium for life insurance or they get discriminated when it comes to their job. These are two things that are quite different. Social discrimination is quite different from something that has direct financial implications like not getting a job or actually having to pay more for your insurance. The increase of reference databases, which is exactly the thing that we're trying to do to improve the interpretation, is exactly the thing that is going against the stream of protecting data. At some point, there will be enough correlations of everything that it will be impossible to say that there's a data set that I can just simply share without a controlled access or without a specific regulatory framework. Which means that it would be a nightmare to do research in that way because we will not be able to share data. We will not be able to share just numbers or quantities. We always have to go through a very, very strict controlled access, which basically makes the point that we need to actually develop that framework. And this data integration will make it also possible to protect the individual. I don't think that it's realistic. I mean, we can think transiently of protecting the individual and protecting the data and so on, but if we really want to make a difference, both at the clinical and the research level, we cannot think of protection in a strict way that we're thinking right now. It's just unrealistic. So the only way to think about it is, first of all, a regulatory framework. As I said, it's the aspect that covers the financial consequences, which are mostly what the legal framework is worried about. And then think about the social framework, because, you know, if somebody's bald or fat or whatever, they don't need to protect that data. That's directly obvious, right? But for other types of data, we feel embarrassed to share it. And maybe that creates also sort of a social issue of, can we actually relieve some of these tensions of what this data means? The more we try to protect it, the more we actually create a social discrimination framework where people feel embarrassed to share that kind of data. So it's actually a very much bigger problem. So at the Genome Center, and together with the platform, the multi-omics platform in Zurich, and together, of course, with many investigators, we have put together and started a discussion regarding all these issues of the ethical framework, as well as how to integrate the sort of the data and research and clinical needs with an ethical framework, develop that, because it's going to come up more and more as we go, and we certainly need that discussion to be as scientific as possible, at the same time taking into account all these social issues. Probably at the end of the day, we need a new legal framework. We need to challenge the legal system than just simply trying to respond and adapt to whatever legal framework exists. So that was sort of my side of things. Thank you very much.